Very, very cool.
Yeah.
All right. So can you guys see the cell respiration slide? Yeah? Okay. All righty. You'll notice that there are captions down here. If the internet gets slow, the captions will stop. Um, if I freeze, sorry. Usually I can tell because I can see fans in the background of the people that are watching. They'll stop. So I know that I'm frozen and I usually try and wait. But again, if you have a question or you need something, feel free to unmute yourself and let me know, okay? Alrighty. So 2.8 is really about cell respiration. But remember, cell respiration and photosynthesis are complementary reactions. They're basically the same process, forwards and backwards. And the purpose of cell respiration is to release energy that's stored in glucose and convert it into ATP. ATP is a molecule that stores energy that your cells can use. So we eat food and we get energy in the form of glucose, but your cells can't use glucose by itself as energy. So you can think of it as like if you ever travel to a foreign country and you have to convert your money, like if you're in Europe, you have to convert your money to euros, or if you're in Canada, you have to like change your money over to Canadian dollars instead. Cell respiration is really the purpose of changing currency. It's changing energy that's stored in glucose to energy that's stored in ATP so that your cells can use it, so they can spend it. Photosynthesis, on the other hand, is the process of capturing energy from the sun, combining it with carbon dioxide and water to produce glucose, right? So on the top here, this is photosynthesis, right? And the job is to capture energy and store it in glucose. And respiration is really the opposite. Respiration is taking that molecule, adding oxygen, and splitting it back into its original components of carbon dioxide and water and releasing that energy in the form of ATP so that your cells can use it. All right, when you're ready, give me a thumbs up. Bella's ready. Quentin's ready. Savannah's ready. Good. Good. Okay. I think we're ready. Alrighty. Pim's good. Brooke's good. Good deal. Drit's ready. All right. So understanding number one, cell respiration, whether we're talking about aerobic or anaerobic, is the controlled release of energy from organic compounds like glucose to produce ATP, adenosine triphosphate. So that's our understanding. So let's break that down and really talk about what that means. So because we're breaking down glucose, right, this is a catabolic reaction. Think all the way back to topic 2 when we talked about catabolic breaking down and anabolic building up, and we talked about all those different classes of molecules. The reason we talked about all that stuff first is because now we're going to start using those concepts and applying them. So the process of cellular respiration is catabolic because it's breaking down glucose to produce ATP. And then your cells use that ATP as an energy source for other chemical reactions. Understanding number two, ATP from cell respiration is immediately available as a source of energy for the cell for processes like active transport, protein synthesis, whatever your cell's job is, right? So if your cell's job, if you are a pancreatic cell and your job is to produce insulin, now you have a molecule that stores energy in a form that you can use and you can pump out a bunch of insulin. Or you can power cellular transport, like active transport. You can also use ATP for condensation reactions, dehydration synthesis, to build compounds, right? Or to move stuff. Those are the major things that we use ATP for. Building and moving. Good. Quentin's ready to go. Good. Okay. So let's talk about ATP for a minute. So ATP is adenosine triphosphate. So adenosine right here. We also have a ribose molecule right here. Triphosphate. 1, 2, 3. There's 3 phosphate molecules. ATP has a high energy content. And all that energy is stored right here in this chemical bond. All chemical bonds are is stored energy, right? So the energy that we use when we use ATP is stored right here. We release that energy by breaking ATP into ADP, and that energy is released. So you can think of ATP as like your cell phone when it's all the way charged, and ADP, because that bond is broken and now we only have 2 phosphates, it's like your cell phone on low battery. So it's low energy. Now, you can convert that ADP back into ATP by putting another phosphate back on and storing energy in it. So ATP and ADP can be recycled. Hey, Matt.
Miss Sanford, I apologize. I was unable to find the Zoom link, so it took me a minute, and I didn't see your remind because my notifications were off. So I'm very sorry for being late.
That's all right. No worries. We're doing lecture 2.8. So if you go to topic 2 in the modules and scroll down to 2.8, that's what we're doing. The PowerPoint is up there, so you can always go back. And I'm recording the lecture too, so I'll post the recording on there for you.
All right. Yes, ma'am. Thank you.
Yep. Alrighty. How do you feel about ADP and ATP? Could you do a compare and contrast? One has 2 phosphate, one has 3. One is high energy, one is low energy. They can both be reused. They're both molecules that store energy. They're different forms of the same molecule. All right, if you're ready, give me a thumbs up. Good. Okay, looks good. All right, so this slide is basically just the same thing. Here you can see the molecular view of adenosine triphosphate. So here's our adenosine, here's our ribose molecule, and here are our 3 phosphates, 1, 2, and 3. And it's the universal energy carrier. So active transport, reproduction, movement, muscle contractions, protein synthesis. We kind of said all that already, right? These are the different things that we use adenosine triphosphate for. So there's our phosphates. Now, the hydrolysis of ATP, right? Hydro means water. Lysis means to break. Hydrolysis. This is a catabolic reaction. This is that same reaction that we just looked at last slide. So you have ATP, you add water, it breaks that phosphate off, and it releases energy. So why do you get thirsty when you're expending a lot of energy? Because you need water to break that ATP down and release that energy so you can use it. Here we have that same diagram. It's just a little easier to see because we don't have the molecular structure there. It's kind of a simplified diagram. So we're going to add water, break that phosphate off, and we release that energy. And it's that heat and that energy that drives cellular processes, like active transport. Okay, so now we're going to start getting into the nitty gritty. So, understanding number 3, anaerobic respiration gives a small yield of ATP from glucose. Glycolysis, which is the breakdown of glucose by enzymes, releases energy and creates pyruvate. So, anaerobic respiration is basically fermentation. And this is an overview of the entire system. So we'll go through the system step by step. But this is an overview. The first step is glycolysis. Glyco, glucose, lysis, breakdown. Glycolysis is the breakdown of glucose. And it releases two molecules of ATP, and it creates a compound known as pyruvate. And I'm going to show you how all this happens. This is just an overview. So take a minute, write these steps down, and then we'll go through it step by step and I'll show you what these steps are. I'll give you a couple minutes to write that. All right, you ready? Or you need another minute or two?
I need one more minute.
Okay. You guys give me a thumbs up when you're ready. I'm happy to wait. Bella's good. Shane is good. Matt's good. Priya's good. Savannah's good. Quentin's good. Pim's good. So let's go through this step by step. Some of you might remember this from freshman year because we did this freshman year too. So let's talk about glycolysis. So here we have a cell, right? Here we have a mitochondria in the cell. We have the nucleus. We have a molecule of glucose. It's got 6 carbon rings. And we have 2 molecules of ATP. So glycolysis happens in the cytoplasm. And we use these 2 ATP molecules and some enzymes to break down this ring of glucose. So there's our first ATP. We're going to use some enzymes to break that ATP along with some water. And then we're going to use our second molecule. Now, this ATP in reality, it doesn't disappear. It just becomes ADP, and it waits around to be recharged. Now, the result is 2 molecules of pGal. Now, you're not going to have to know this. I'm just showing you. I'm a visual person, so I need to see the way things happen in order to understand it. Like that last slide that like listed the process, that's cool, but like that means nothing to me. I have to like see what's going on. So you're not going to have to actually like draw these phosphoglyceraldehyde or pGal molecules. Just know that when you first break down the glucose, there's an intermediary step and it breaks down into these molecules. It's important because these phosphates are added to make this a stable molecule. Remember how we talked about how carbon needs 4 bonds to make it stable? Right? So that's why you need these phosphates. That's where they go. You break down the ATP and those phosphates are bonded to these molecules to help make it stable. And we're going to add some phosphates on the other side to make sure that that molecule is stable. So now we have these 2 molecules here. Now, NADH is an energy carrier molecule, just like ATP. But it stores energy in the bonds from the hydrogen it steals. So these guys are like cookie monsters. Now remember, this is a glucose molecule, and our formula for glucose is actually C6H12O6. Now, in the picture that I just had a bunch of circles with C's in it to represent a 6 carbon molecule. But remember, there's still hydrogen in there, and there's oxygen in there too. So these NAD molecules are going to steal some of those hydrogens. They're like cookie monsters, stealing cookies from the cookie jar. And they're going to bring that hydrogen to the mitochondria. So we take NAD and we turn it into NADH. Do some of you guys remember this from freshman year? Priya's nodding her head. This is like the exact same slides I use in everything for just this part. All right, so after that NADH is created, we're going to create 4 molecules of ATP by pulling these phosphates back off. So here's 4 molecules of ADP that are waiting around to be recharged. They're going to pull those back off.
Miss Sanford?
Yeah.
I think I missed something. Where does the ADP, like the additional ones, come from again?
They have been waiting around in the cell, waiting to be recharged. So they were used in some sort of process, whether they used to be ATP, and then the cell used them to like transport some molecules out. So they're like low battery. So they're waiting in the cell to be recharged.
Right, but there was only 2 originally. So are these just like previous ones?
Mm-hmm.
Oh, okay. And
Yeah.
it was like left over, but not from this most recent one.
Right. So when we started this process, we started with 2 ATP, meaning that was energy that was waiting to be spent. And we spent that energy and added those phosphates to help break that carbon molecule down. Now, the 4 ADP, 2 of those could have been the same 2 that we just used, plus there's another 2 that are waiting to be recharged. Because remember, this is happening in like all different places around the cell.
Yeah. This is a dumb question, but
No.
what about the very, very first process? Where did the additional ones come from?
Ah, that is a good question. So, my best guess for that is, remember during mitosis, right? You have the division of cytoplasm. So I'm thinking there's a bunch of ATP and ADP in that cytoplasm from that parent cell. And then when it splits, each new cell gets half of what's there. And then the cycle just continues.
Okay, that makes sense.
That's a good question.
Okay, thank you.
You're welcome. So at the end of this process, we have officially created 4 ATP and 2 pyruvate molecules. Now, these pyruvate molecules are important. They, along with those NADH molecules that we created, are going to go to the mitochondria. Well, that's for aerobic respiration. So, glycolysis, long and short. Are you going to have to document every single step? No. What do you need to know? You need to know the starting products and the finishing products, right? So, one more time. We started with 1 molecule of glucose and 2 ATP. We use those ATP to split that carbon molecule, that glucose molecule apart. We're going to use some enzymes to add phosphates to make these stable molecules. NAD is going to come steal some hydrogens off of this molecule and store some energy in that bond, right? So these are also going to contain, there's energy. All chemical bonds are stored energy, right? So by bonding that hydrogen to this NAD molecule, it's storing energy in that bond, and it's going to take it to the mitochondria. Now, we have these phosphates that are at the end of these carbon molecules, right? So here are 4 ADP that are waiting to be recharged. We're going to recharge them by pulling those phosphates off. So we end up with 2 molecules of pyruvate, 4 ATP, and 2 NADH. Now, I think in the slide a minute ago, I said we only create 2 ATP, but clearly we created 4. So why did I tell you a few minutes ago that we only created 2?
Doesn't it use 2, and so it's a net gain?
Right. So we had to spend 2 to make 4. So our net gain is 2. Very good. Okay. So if we don't have oxygen, or say you're a prokaryotic cell and you don't have a mitochondria, right? There's no way to do aerobic respiration. So 2 things can happen. There's alcoholic fermentation and there's lactic acid fermentation. We're going to talk about alcoholic fermentation first. And this occurs in yeast, right? And this is how we make beer and wine and alcoholic beverages. It's actually also how we bake bread. The results of alcoholic fermentation are ethanol, which is alcohol, and carbon dioxide. And we use that chemistry to create good stuff. So here's our glucose, our sugar, right? And it produces ethanol, which is an alcohol, and carbon dioxide. So this is application 1, ethanol alcoholic fermentation. Use of anaerobic cell respiration in yeast to produce ethanol and carbon dioxide. All right, you ready? Should we pause for a minute? You guys want to go get a drink and like stretch? Yeah? Okay, let's pause. We're going to pause for just a few minutes. Let's say 5-minute break, and then we'll come back. Okay?
Hey, Miss Sanford.
Yeah.
I saw that because I'm looking at the PowerPoint on Canvas as well. Are we going through all 54 slides today?
Nah, no way. Probably not.
Okay.
I
was wondering.
Yeah, no. That would like fry everybody's brain. We're not doing that. We'll take a break. I think we'll probably finish alcoholic fermentation. We'll probably do lactic acid fermentation, and then we'll probably stop.
Got it.
Okay? All right, pause. You can turn your cameras off if you want. Let's see. It is 1:22, so at 1:27 we'll come back.
So, Miss Sanford, for this assignment, do you not want to submit it until we're all the way done with 2.8?
No, you can just whatever we get done today.
Okay, got it.
Okay?